The vessel network at the back of the eye is where the retina, a part of the nervous system, is located. It can be easily visualized in a non-invasive way since the eye is transparent. In fact, accumulating evidence shows that ocular symptoms always precede the traditional diagnosis of central nervous system pathologies such as Alzheimer's disease. Therefore, in vivo morphological and functional imaging of retinal structures provide a non-invasive approach to not only understand eye diseases, but also decipher the mechanisms behind neurodegenerative disorders in the central nervous system. The MRI is currently one of the most popular imaging techniques to this day, but it cannot capture the retinal capillaries as they are too small. That is why, most of the time, microvascular diseases are spotted too late when the patient experiences loss of vision or blindness. Therefore, a new optical imaging technique originally used in astronomical telescopes called adaptive optics, or AO for short, is employed to improve the resolution for retinal imaging. AO imaging systems use active optical elements to compensate for aberrations in the optical path between the object and the camera. So in this presentation, we will review two of the advances that were made possible with AO imaging, as well as discuss applications and future prospects for clinical imaging.
Now, let's talk about the objectives of the project. So the first objective is to create an adaptive optics imaging system that allows the high-resolution visualization of the retina, the optic nerve, the microglial structures, and their dynamic behavior, as well as the retinal blood vessels. The second objective is to create a non-invasive way of studying the full spectrum of single blood cell flow in humans and microvascular diseases, and therefore facilitate the diagnosis of central nervous system disorders at an early stage.
The two-photon excitation fluorescence microscopy, or for short, TPEFM, is one of the new optical imaging tools that have been developed. It employs a powerful technique that monitors the dynamic behavior of the chemical components of tissues. But first of all, to understand how neurological disorders work, we need to have a basic understanding of the optic nerve, which is connected to the retina. The optic nerve is composed of the axons of RGCs, which are the retinal ganglion cells, and it transmits visual information from the retina to the thalamus and the midbrain. Axonal degeneration in RGCs have been regarded as one of the most important early signs of neuropathy, which is the damage or dysfunction of one or more nerves, and it may result in devastating consequences such as progressive RGC death and irreversible vision loss. Furthermore, neuroinflammation, which is the chronic inflammation of nervous tissue typically associated with neurodegenerative diseases, is largely regulated by microglia, the resident immune cells in the central nervous system. The microglia are highly sensitive to subtle alterations in the microenvironment, which retinal injury and neuroinflammatory diseases would cause. So, in order to study the early onset and development of retinal diseases, there must be in vivo imaging and analysis of microglial structures as well as their dynamic behavior. So this has only been tested on mice, but compared to the sensorless AO technique, the AO-TPFM method can usually achieve faster AO correction and visualize more clearly the dynamic biological structures and processes in the retina. The imaging techniques were performed on a mouse, and its eye was injected with a substance to label retinal neurons with green fluorescent protein. The results showed that with AO correction alone, the fluorescence images are severely blurred, as you can see in the images in the first row. But after full correction, the images reveal the fine structures of neurons and processes in different retinal layers. In addition, fine structures of microvascular and neuronal networks in the retina can be visualized simultaneously, which is shown by the second image, which is also much clearer than the first image. So as you can see, after AO correction, the AO-TPFM allows the identification of early microglial activation in response to pathogens. Given that microglial activation often precedes reactions of other cell types in the central nervous system, the non-invasive detection of microglial morphologies by AO-TPFM may facilitate the diagnosis of central nervous system disorders at an early stage. By imaging the dynamics of various retinal structures, With high spatial and temporal resolution, this technique offers great opportunities to disclose the mechanisms of eye diseases and evaluate therapeutic strategies with high fidelity.
As we have mentioned previously, current imaging techniques do not correct the many aberrations which blur microscopic retinal capillaries and single blood cells flowing within. This limits early evaluation of microvascular diseases that originate in capillaries. Using a system called Adaptive Optics Scanning Light Ophthalmoscope, or AOSLO, we are able to report single-cell velocity and flow rate in the smallest to largest vessels in a living mouse retina. This was enabled by AOSLO imaging coupled with ultra-fast camera capture to non-invasively image blood cells without foreign contrast agents. Paired with an automated analysis workflow, it enables velocity measurement of hundreds of thousands of blood cells per second. Together, these developments provide a new, automated and label-free way of determining vascular perfusion and flow dynamics in the mammal retina in conditions of health and disease. This imaging technique allows recovery of mice after anesthesia, allowing them to be returned to their cages, thus enabling in vivo imaging in the same mouse. In other words, the mice can still live without any deficits. Vascular maps obtained during imaging provide the ability to track the same retinal location in the same animal over weeks to months, which is critical for diagnosing progression and treatment efficacy in the mouse models of diseases like diabetes. Moreover, measurements were performed using safe infrared light levels, to which the eye is insensitive. There has also been some early data of the same technique applied to humans, with automated velocity measurements performed in retinal vessels of intermediate size. Now, let's actually take a look at the images that were taken in mice. So here we have an image of a capillary, and we can see the single red blood cells within. So in figure B, we can see the capillary, and in C, all those little circles are single blood cells. Next, this image shows how they were able to calculate the velocity of blood flow with a computer algorithm. In the first picture, there's a space-time image of a 23.3 micrometer arteriole. In the yellow box, we have the region of interest. Using the radon transform, the computer algorithm, the slope of blood cell streaks was measured. And then, using multiple calculations, the velocity of the blood flow within various blood vessels was found.
Now, we won't go into too much detail about how exactly the two systems work because it is way too complicated, but we will briefly explain their setups. The AOTPFM method uses direct wavefront measurement of a nonlinear fluorescent guide star for functional retinal imaging in living mice. It is composed of two lasers that are combined using a polarizing beam splitter. That laser beam is then reflected by the DM, which is a deformable mirror, and demagnified to match the scan mirrors. One of those mirrors is then relayed to the ETL, which is then relayed to the mouse cornea. This system was designed so that it can be easily incorporated into standard microscope systems for in vivo retinal imaging through the mouse pupil. For two-photon fluorescence imaging, the fluorescence emission signals are reflected by dichroic mirrors and then separated into green and red channels. Appropriate filters are placed in front of the photomultiplier tubes, the PTM, to select particular wavelength ranges of detection. And for wavefront sensing, the emitted fluorescence signal retraces its path back through the scanning mirrors, and it is then picked up by the custom-built Shack-Hartman wavefront sensor. And then finally, subcellular retinal imaging through the pupil of the mouse eye will be achieved.
The AOSLO system, which stands for Adaptive Optics Scanning Light Ophthalmoscope, is another method of imaging. So this picture is not the exact same model that was used in the experiment, but it provides a good idea of how it works. So it uses three light sources. The first one is acting as the wavefront sensing beacon. The second one provides near-infrared reflectance imaging. And finally, the third one is used as the excitation source for sodium fluorescence. These three light sources are then combined into one single common source with dichroic mirrors, and then relate to the eye using reflective optics through a series of 5 afocal telescopes. The field of view can be easily altered to vary between 0.5 and 6.2 degrees, and all images were digitized into 640 by 480 pixels. There is also a large stroke high-speed deformable mirror, which corrects the aberrations of the eye. The deformable mirror also provides programmable defocus control, which allows the focusing of the imaging beam on a layer of interest in the retina without using additional optics. This imaging system was paired with an algorithm that analyzed the flow of hundreds of thousands of blood cells. Although the two models are very different, they still possess some similarities, such as the use of wavefront sensors, excitation lasers, and deformable mirrors. Though they are both very effective, we thought of an innovative concept that would involve developing a model that would combine the two systems. Therefore, it would make an improved model that could be used on the human eye instead of mice, in a safe and non-invasive way. Theoretically, if it were possible, it would satisfy both our objectives, since it would allow optimal visualization of not only the optic nerve and the microglia, but also the retina and its blood vessels. To conclude, it has long been proposed that the eye may serve as a window to the brain. Current imaging techniques like the MRI do not offer a clear image, especially at the capillary level. Vascular diseases like diabetes, atherosclerosis, and hypertension are known to impact the microscopic vessels of the body. Using adaptive optics allows for a non-invasive way of studying the full spectrum of single blood cell flow in humans and microvascular diseases. All this will allow the development of new treatments as it can offer a clearer view of the mammalian eye.